Hello everyone, my name is Stephanie Mata-Bonilla. This is a poster of my master thesis entitled Characterization of Messenger RNAs and Nucleic RNAs of Botryococcus brownie recipe strain Showa. First of all, Botryococcus brownie is a microalgae that belongs to the chlorophyll class. At a day, it is the only organism with a capacity to produce hydrocarbons. Its growth rate is slow and its knowledge is limited. That is why it's important to study how biological processes are regulated in the browning cells to find alternatives that allow us to accelerate its growth and obtain a greater accumulation of hydrocarbons. For this study, my project was cultured under normal growth conditions and conditions of carbon dioxide excess of 1.5% of carbon dioxide. After 22 days of culture, the biomass was collected by filtration. When we made a process of RNA extraction, the RNA quality was checked by bioanalyzer, cubed and electric forensics. Okay, samples with best quality were selected for sequencing, and from sequencing we obtained pair and electron-specific copy DNA libraries that was used to make a bioinformatic analysis. Okay, the bioinformatic pipeline was this. Using chemomatic libraries were processed to remove sequencing adapters. The quality of libraries was verified and the data was aligned against microarray reference genome and transcriptome were assembled in a guided manner. We verified the quality of the assembled transcriptome and it was used for the annotation of long-non-coding RNAs and their classification according to genomic coordinates. Also, the transcriptome was used to obtain gene counts that allowed us to perform a differential expression analysis and a genome pathology analysis. As a result, well, first, we had the opportunity to annotate and classify long-non-coding RNAs for the first time in this microalgae. We used two methodologies. With coding potential calculator, we found 1,391 long-non-coding RNAs, and a strict pipeline described in Kabil et al. In 2011 also was used. With this, we annotate 867 long-non-coding RNAs. This last methodology allowed us to identify potential long-non-coding RNAs in a most robust way. These long-non-coding RNAs presented lengths similar to those reported in macroalgae such as Fernandez-Valverde renardi and smaller than those reported in plants like Arabidopsis thaliana. The 867 long-non-coding RNAs were classified according to the genomic coordinates of microalgae genome. It was found that most of them are transcribed in a sense manner, but in addition we found proximal and long intergenic long-non-coding RNAs. And antisense long-non-coding RNAs also cause NOTs and intronic long-non-coding RNAs that lost in smaller amounts. Around 368 coding genes and 14 long-non-coding RNAs were differentially expressed in low carbon dioxide conditions. Also, 177 coding genes and 8 long-non-coding RNAs were differentially expressed in high carbon dioxide conditions. It was possible to us to associate differentially expressed genes with biological processes in both growth conditions, biological processes, and molecular functions. In general terms, it was found that in carbon dioxide excess, the metabolism of the microalgae increases, and it is clearly observed that we could find a higher rate of carbon dioxide fixation compared to low carbon dioxide conditions. We conclude that the long ncRNAs, differentially expressed in this microalgae in high concentrations of carbon dioxide, could be involved in the regulation of carbon dioxide transport and fixation. However, more studies and experimental validation need to be done. Also, maybe in a carbon dioxide excess, there will be an increase of the synthesis of hydrocarbons, which will mean an alternative to increase its production and scale it for future commercial purposes. I want to say thank you to Conacyt to provide us the funds to make this work possible, and also thank to Women's and Bioinformatics staff to give me the opportunity to share my work with you. Thanks for seeing this video, and bye bye.